Welcome back to Chapter 4. This is Section 2, Proteobacteria. Learning objectives. Describe the unique features of each class within the phylum Proteobacteria, alpha, beta, gamma, delta, and epsilon proteobacteria. Give an example of a bacterium in each class of Proteobacteria. In 1987, the American microbiologist Carl Woese, who lived from 1928 to 2012, suggested that a large and diverse group of bacteria that he called purple bacteria and their relatives should be defined as a separate phylum within the domain Bacteria based on the similarity of their nucleotide sequences in their genome. This phylum of gram-negative bacteria subsequently received the name Proteobacteria. It includes many bacteria that are part of the normal human microbiota, as well as many pathogens. The Proteobacteria are further divided into 5 classes: Alphaproteobacteria, Betaproteobacteria, Gammaproteobacteria, Deltaproteobacteria, and Epsilonproteobacteria. Alphaproteobacteria. The first class of Proteobacteria is the Alphaproteobacteria. The unifying characteristic of this class is that they are oligotrophs, organisms capable of living in low-nutrient environments such as deep ocean sediments, glacial ice, or deep undersurface soil. Among the Alphaproteobacteria are 2 taxa, Chlamydias and Rickettsias, that are obligate intracellular pathogens, meaning that part of their life cycle must occur inside other cells called host cells. When not growing inside a host cell, Chlamydia and Rickettsia are metabolically inactive outside of the host cell. They cannot synthesize their own adenosine triphosphate, ATP, and therefore rely on cells for their energy needs. Rickettsia species include a number of serious human pathogens. For example, R. rickettsii causes Rocky Mountain spotted fever, a life-threatening form of meningoencephalitis, inflammation of the membranes that wrap the brain. R. rickettsii infects ticks and can be transmitted to humans via a bite from an infected tick. Another species of Rickettsia, Rickettsia prowazekii, is spread by lice. It causes epidemic typhus, a severe infectious disease common during warfare and mass migrations of people. R. prowazekii infects human endothelium cells, causing inflammation of the inner lining of blood vessels, high fever, abdominal pain, and sometimes delirium. A relative, R. typhi, causes a less severe disease known as murine or endemic typhus, which is still observed in the southwestern United States during warm seasons. Chlamydia is another taxon of the Alphaproteobacteria. Members of this genus are extremely resistant to cellular defenses, giving them the ability to spread from host to host rapidly via elementary bodies. The metabolically and reproductively inactive elementary bodies are the endospore-like form of intracellular bacteria that enter an epithelial cell where they become active. This figure illustrates the life cycle of Chlamydia. Chlamydia trachomatis is a human pathogen that causes trachoma, a disease of the eyes often leading to blindness. C. trachomatis also causes the sexually transmitted disease lymphogranuloma venereum. This disease is often mildly symptomatic, manifesting as regional lymph node swelling, or it may be asymptomatic, but it is extremely contagious and is common on college campuses. Here is a table that summarizes some common Alphaproteobacteria. Unlike Alphaproteobacteria, which survive on a minimal amount of nutrients, the class Betaproteobacteria are eutrophs, or copiotrophs, meaning that they require copious amounts of organic nutrients. Betaproteobacteria often grow between aerobic and anaerobic areas. like in the mammalian intestines. Some genera include species that are human pathogens, able to cause severe, sometimes life-threatening disease. The genus Neisseria, for example, include the bacteria in gonorrhea, the causative agent of the sexually transmitted infection gonorrhea, and Neisseria meningitidis, the causative agent of bacterial meningitis. Neisseria are cocci that live on mucosal surfaces of the human body. They are fastidious or difficult to culture. And as an aside, I can verify that one. They are difficult to culture. And they require high levels of moisture, nutrient supplements, and carbon dioxide. Also, Neisseria are microaerophilic, meaning that they require low levels of oxygen. For optimal growth and for the purposes of identification, Neisseria species are grown on chocolate agar, which is agar that is supplemented by partially hemolyzed red blood cells, which you can readily achieve by boiling your blood before you add it. Their characteristic pattern of growth and culture is diplococcal, pairs of cells resembling coffee beans. The pathogen responsible for pertussis, whooping cough, is also a member of Betaproteobacteria. The bacterium Bordetella pertussis from the order Burkholderiales produces several toxins that paralyze the movement of cilia in the human respiratory tract and directly damage cells in the respiratory tract, causing a severe cough. This table summarizes the characteristics of important genera of Proteobacteria. So this includes Bordetella, Burkholderia, Leptothrix, Neisseria, and Thiobacillus. Clinical focus, Part 2. When Marcia finally went to the doctor's office, the physician listened to her breathing through a stethoscope. He heard some crepitation, a crackling sound, in her lungs, so he ordered a chest radiograph and asked the nurse to collect a sputum sample for microbial evaluation and cytology. The radiologic evaluation found cavities, opacities, and a particular pattern of distribution of abnormal material. What are some possible diseases that could be responsible for Marcia's radiograph results? Gammaproteobacteria. The most diverse class of gram-negative bacteria is Gammaproteobacteria, and it includes a number of human pathogens. For example, a large and diverse family, Pseudomonaceae, includes the genus Pseudomonas. Within this genus is the species Pseudomonas aeruginosa, a pathogen responsible for diverse infections in various regions of the body. Pseudomonas aeruginosa is a strictly aerobic, non-fermenting, highly motile bacterium. It often infects wounds and burns, can be the cause of chronic urinary tract infections, and can be an important cause of respiratory infections in patients with cystic fibrosis or patients on mechanical ventilators. Infections by Pseudomonas aeruginosa are often difficult to treat because the bacterium is resistant to many antibiotics and has a remarkable ability to form biofilms. Other representatives of Pseudomonas include the fluorescent, glowing bacterium, Pseudomonas fluorescens, and the soil bacteria, Pseudomonas pudita, which is known for its ability to degrade xenobiotics, substances not naturally produced or found in living organisms. The Pasteurellacea also include several clinically relevant genera and species. This family includes several bacteria that are human and or animal pathogens. For example, Pasteurella hemolytica causes severe pneumonia in sheep and goats. Pasteurella multocida is a species that can be transmitted from animals to humans through bites, causing infections of the skin and deeper tissues. The genus Haemophilus contains two human pathogens, Haemophilus influenza and Haemophilus ducreyi. Despite its name, Haemophilus influenza does not cause influenza, which is a viral disease. Haemophilus influenza can cause both upper and lower respiratory tract infections, including sinusitis, bronchitis, ear infections, and pneumonia. Before the development of effective vaccination, strains of Haemophilus influenza were a leading cause of more invasive diseases like meningitis in children. Haemophilus ducreyi causes the STI known as cancroid. The order Vibrio-Nales includes the human pathogen Vibrio cholera. This comma-shaped aquatic bacterium thrives in highly alkaline environments like shallow lagoons and seaports. A toxin produced by Vibrio cholera causes hypersecretion of electrolytes and water in the large intestine, leading to profuse watery diarrhea and dehydration. Vibrio parahemolyticus is also a cause of gastrointestinal disease in humans, whereas Vibrio vulnificus causes serious and potentially life-threatening cellulitis, infection of the skin and deeper tissue, and blood-borne infections. Another representative of Vibrio-Nales, Vibrio fischeri, engages in a symbiotic relationship with squid. The squid provides nutrients for the bacteria to grow, and the bacteria provide bioluminescence that protects the squid from predators. The genus Legionella also belongs to the Gammaproteobacteria. Legionella pneumophila, the pathogen responsible for Legionnaire's disease, is an aquatic bacterium that tends to inhabit pools of warm water, such as those found in the tanks of air conditioning units in large buildings. Because the bacteria can spread in aerosols, outbreaks of Legionnaire's disease often affect residents of a building in which the water has become contaminated with Legionella. In fact, these bacteria derive their name from the first known outbreak of Legionnaire's disease, which occurred in a hotel hosting an American Legion Veterans Association convention in Philadelphia in 1976. Enterobacteriaceae is a large family of enteric intestinal bacteria belonging to the Gammaproteobacteria. They are facultative anaerobes and are able to ferment carbohydrates. Within this family, microbiologists recognize two distinct categories. The first category is called the coliforms, after its prototypical bacterium species, Escherichia coli. Coliforms are able to ferment lactose completely with the production of acid and gas. The second category, non-coliforms, either cannot ferment lactose or can only ferment it incompletely, producing either acid or gas, but not both. The non-coliforms include some notable human pathogens, such as Salmonella species, Shigella species, and Yersinia pestis. Escherichia coli has perhaps been the most studied bacterium since it was first described in 1886 by Theodore Escherich, 1857 to 1911. Many strains of E. coli are in a mutualistic relationship with humans. However, some strains produce a potentially deadly toxin called Shiga toxin, which perforates cellular membranes in the large intestine, causing bloody diarrhea and peritonitis, inflammation of the inner linings of the abdominal cavity. Other E. coli strains may cause traveler's diarrhea, a less severe but very widespread disease. The genus Salmonella, which belongs to the non-coliform group of Enterobacteriaceae, is interesting in that there is still no consensus about how many species it includes. Scientists have reclassified many of the groups they once thought to be species as serotypes, also called serovars, which are strains or variations of the same species of bacteria. Their classification is based on patterns of reactivity by animal antisera against molecules on the surface of the bacterial cells. A number of serotypes of Salmonella can cause salmonellosis, characterized by inflammation of the small and large intestine, accompanied by fever, vomiting, and diarrhea. The species Salmonella enterobacterica, serovar typhi, causes typhoid fever, with symptoms including fever, abdominal pain, and skin rashes. So this table summarizes the characteristics of important genera of Gammaproteobacterium. Deltaproteobacteria. The Deltaproteobacteria is a small class of gram-negative proteobacteria that includes sulfate-reducing bacteria, SRBs. so named because they use sulfate as the final electron acceptor in the electron transport chain. Few SRBs are pathogenic. However, the SRB desulfovibrio orale is associated with a periodontal disease of the gums. Deltaproteobacteria also include the genus Betalovibrio, a species which are parasites of other gram-negative bacteria. Betalovibrio invades the cells of the host bacterium, positioning itself in the periplasm, the space between the plasma membrane and the cell wall, feeding on the host's proteins and polysaccharides. The infection is lethal for the host cells. Another type of Deltaproteobacteria, a myxobacteria, lives in the soil, scavenging inorganic compounds. Motile and highly social, they interact with other bacteria within and outside their own group. They can form multicellular macroscopic fruiting bodies, pictured here, structures that are still being studied by biologists and bacterial ecologists. These bacteria can also form metabolically inactive myxospores. And again, here is a table that summarizes the characteristics of several important genera of Deltaproteobacteria. Epsilonproteobacteria. The smallest class of proteobacteria is Epsilonproteobacteria, which are gram-negative microaerophilic bacteria, meaning they only require small amounts of oxygen in their environment. Two clinically relevant genera of Epsilonproteobacteria are Campylobacter and Helicobacter, both of which include human pathogens. Campylobacter can cause food poisoning that manifests as severe enteritis, inflammation of the small intestine. This condition, caused only by the species Campylobacter jejuni, is rather common in developed countries, usually because of eating contaminated poultry products. Chicken often harbor C. jejuni in their gastrointestinal tract and feces, and their meat can become contaminated during processing. Within the genus Helicobacter, the helical flagellated bacterium Helicobacter pylori has been identified as a beneficial member of the stomach microbiota, but it is also the most common cause of chronic gastritis and ulcers of the stomach and duodenum. Studies have shown that Helicobacter pylori is linked to stomach cancer. Helicobacter pylori is somewhat unusual in its ability to survive in the highly acidic environment of the stomach. It produces urease and other enzymes that modify its environment to make it less acidic. So here is a table that summarizes the characteristics of the most clinically relevant genera of class Epsilonproteobacteria. Then that brings us to the end of Section 2. Join me next time for Section 3, non-proteobacteria, gram-negative bacteria, and phototropic bacteria. Until then.